Nice to meet you both.
Great to meet you.
Nice to meet you as well. So we were wondering, can we also get like a quick introduction on your background and kind of the work that you currently do and the field that you're in?
Sure. So I sometimes refer to myself as a clinical genomicist and rare disease researcher. So my background is a PhD in genetics, but then a short postdoc, and then I did a fellowship in clinical laboratory genetics that um, gave me the training to direct a genetic testing lab. Um, and then from there, I actually built and ran a genetic testing lab at Mass General Brigham for about 17 years. And during that time, I started getting more involved in NIH funded research programs in genomic medicine. And so kind of got much more back into basic, not basic research, but translational research. And then after 17 years, I ended up moving over to MGH to become the chief genomics officer and also focus more on my rare disease research and other genomic resource programs like the clinical genome resource. And now I co-run Nomad, the genome aggregation database. I also lead a lot of data sharing projects in rare disease, like matchmaker exchange, variant level matching, federated nomad. Um, and I have a lot of sort of side roles, things like I chair the Global Alliance for Genomics and Health, which is a standards building organization. Um, I'm on the board of American Society of Human Genetics. I was on the board of American College of Human Genetics. So I am engaged in the community in a lot of ways to develop sort of standards for how we operate in the clinical genomics space and the resources that we all have to support the work we're doing.
That's amazing. And uh, could you tell us where your interest in biology and genetics first stemmed from?
Well, I grew up in a household where my father was a high school biology teacher, and I spent a lot of time outdoors, and I just loved math and science. And then was particularly fascinated by genetics. Some of it is I'm a very type A personality. I sort of like to organize the world and make the world more efficient and effective. And genetics was this incredibly elegant sort of biological system where, you know, our genes encode RNA, which encodes protein, the sort of central dogma. And then those proteins do different things in cells. And then when you disrupt them through mutation, they stop working and you get a disease. And it was just a really both simple and elegant system where that was tangible to me. And so this fascination with being able to, study the genome, identify disruptions, correlate them with disease, diagnose patients, give them information that was useful for their care and understanding why they had a genetic disorder. It was, something that I felt was of interest and tangible to actually work on.
I think that's also kind of a similar reason why I'm also interested in biology. I find it like fascinating how like DNA, which is so small, like you need it's on a molecular level, and such a small change can impact you in such large ways. So I think that's similar reason why I'm interested as well.
Yes, I completely agree.
Kind of
And
bouncing
then
off what Veer just said. How can two patients with changes in the same gene sometimes have completely different symptoms or disease severity? Completely different. How's that possible?
So there's many different explanations for that. Most of the time, we simply don't know, but there's been enough examples of different things that affect that, and they can range from either environmental influences on, so for example, some very obvious ones. There's a mutation in an RNA gene that leads to uh, genetic hearing loss. And if you're exposed to aminoglycoside antibiotics, then the likelihood that you develop hearing loss is much, much higher than if you don't have this. And it's basically the conformation change of the RNA with this mutation makes it look like the RNA of a bacterium. And so essentially this RNA gets targeted and destroyed and then leads to a worsening phenotype. So that's just like a very concrete example of the interaction between the environment and genetics that could explain why one person doesn't have hearing loss with a mutation and another one does. There are, other factors. Another one would be dilated cardiomyopathy, where the likelihood that your heart and the muscle tissue that's affected by mutations in the sarcomere genes, that can be exacerbated by either being a professional athlete that puts a lot of strain on your heart, or an IV drug user, or even pregnancy. So there's different things that could exacerbate an underlying genetic defect. So those are sort of modifiers in the environment that can influence the expression of genetics. But then there can be other genetic modifiers. So for any gene that's on the non-sex chromosomes, we have two copies. And what's happened, for a dominant disorder that's caused by mutations in one gene, what's happening on the other copy of the gene can influence the severity of the phenotype. So if the other copy of the gene is wild type and it's more highly expressed than the bad copy in one person, and then another person with the same mutation, the bad copy is overexpressed compared to the wild type, that will give different severities of disease based on the amount of bad versus good gene expressed, right? So there's lots of, and then half the time we just don't even know. But those are some examples of things that we've uncovered over time.
Got it. And then speaking on that unknown, especially in rare diseases, there's so much unknown. How do you navigate that personally?
Yes, it's very frustrating, particularly for the patients who, we suspect they have a genetic disorder. They're seeking an answer, and we come back with nothing. That's frustrating. And I think for one of the reasons, I said I was a lab director for 17 years. One of the reasons I kind of switched to research and resource building is that I felt like I'd done everything interesting I could do by spending most of my time in a clinical lab. And I wanted to really spend my time advancing the field so that we could give more complete results back to patients. And so, I kind of basically say, look, today your answer is nothing. We haven't found anything, but we're going to keep looking at your genome. And now we're building automated reanalysis pipelines so that we're constantly, relooking at their genomes. New discoveries are being made, and we go back to genomes we haven't solved and apply these new things to them. So, we're still engaged with our research subjects. They check in time to time. We ask for new samples to run new technologies on them that might help solve and find an answer. So we're never sort of giving up. And I think that's in some ways how I reconcile the lack of answer today.
And for patients, genetic diagnosis can be described as the beginning rather than the end of a patient's journey. So, what can a diagnosis make possible or open the door to that wouldn't be possible before?
Right. So, um, there's a lot of diseases that can evolve over time. And so, the question of, all right, my child has developmental delay and, currently can't speak, or, there's different things. And parents, will come and say, well, are they going to get worse? Could they get better? Are other things that I haven't seen today going to start? Are they going to start getting seizures? Like what's the future hold? And so once you have a diagnosis, then you have a set of patients who also have that diagnosis, who range in age from younger to older than that individual, and it gives them a glimpse into the future of and other families to talk to. Well, how have you addressed this issue or that issue? And so it's like you all of a sudden have a whole community, and there's a lot of patient support organizations built around specific genes or diseases, and these families find they have a home and a place to ask questions of, and things to learn from that experience. And, there's also now the ability to think about interventions that may be um, management, where they know what treatments have worked or not worked. The ability to think about genetic therapeutics. You can't do that if you don't know the genetic etiology. So, the other thing that we find is just having an answer, even if you get no more information, can be incredibly beneficial to these families who have spent their life thinking, oh, it was that glass of wine I had during my second trimester. That's probably why it was my fault, or, like blaming themselves, and getting an answer and making it clear, no, that wasn't your fault. You had no control over this, can be incredibly heartening to these families, right?
And speaking on, I think you briefly touched on like patient advocacy groups and the importance of connecting with other people that share a similar condition. So, what do you think makes rare disease specifically so dependent on collaborations between clinicians, researchers, patients, and these advocacy groups?
Right. So, rare diseases, the name is a little complex. As a collection, rare diseases aren't rare. They actually afflict, 1 in 10 individuals. Um, but individually, many of them are rare, and they are highly variable in their from. So there's about, I don't know, about 150 to 200 diseases that make up over 80% of all of the individuals with rare disease. And these are, things you might have heard of before, like cystic fibrosis, hypertrophic cardiomyopathy, and sickle cell disease, and hereditary breast cancer, and, the ones that are more common on average than most rare diseases. But then there is this incredible long tail of very, very rare diseases, um, thousands and thousands of them that, are fewer than, most of which are fewer than 1 in 1 million. And so when you've got a disease that's afflicting 1 in 1 million people, then you, if you're going to learn anything about that disease, you need to find those handful of people with that disorder so that you can learn whatever you can from that small subset of people. So that is really why it's so important that we share data, that we collaborate, and we can collectively learn together. The other thing that we've done is we have this project that Sam Baxter and my team leads called Genie, and it's a tool basically to help figure out the prevalence of rare disorders, because a lot of these patient advocacy groups that are trying to, collect their patients together and then advocate for pharmaceutical biotech companies to develop treatments for their disease, they won't, those companies won't invest if they don't have a large enough market. And so trying to figure out the prevalence of a given disorder can be incredibly important to advocate for treatment development for a given disorder. So, um, that's another, really important thing in terms of bringing people together.
You touched upon the economic side a little bit. As genomic sequencing has become pretty much dramatically less expensive over the past few decades, how has that change affected what is realistically possible in clinical medicine?
Um, it's allowed us to take what we often call a genotype first approach. So, early on when it was really expensive to sequence, you would just sequence the gene or maybe a small set of genes that were correlated to the phenotype you observed in your patient. So, a patient who had cystic fibrosis clinically based on sweat chloride tests, based on, lung disease, pancreatic insufficiency, various factors, would then get a test and sequence variants in the CFTR gene. Well, if you already know the patient has cystic fibrosis, then that's not all that, it's not going to change how you care for that patient. You already know they have cystic fibrosis. The CFTR test may be useful for carrier or for um, pregnancy planning and the risk that you're going to have a second child with that, but it's not really going to change what you do for that first child. You already know their clinical diagnosis. In contrast, there's a lot of disorders where you can't predict which gene it is, and it becomes quite important, and you don't know what set of genes or what gene to test based on the phenotype. They're particularly like neurodevelopmental disorders can be very difficult to try to figure out what the gene might be because they're non-specific symptoms like global developmental delay and failure to thrive and hypotonia and macrocephaly and seizures and this and that. So by taking a genotype first approach where you run a genome and you don't have to know in advance which genes to look at, the likelihood you can find a genetic diagnosis goes way up. And so that has been, game changing, particularly in certain fields like neurodevelopmental disorders, primary immunodeficiencies, various things that are, the phenotypes can be quite complex to try to anticipate in advance what the genetic etiology might be.
Got it. And how do you think we should evaluate the economic value of a diagnosis when the immediate outcome isn't or may not be a treatment, but rather more of an explanation or better care, or even the ability to connect the patient to future research?
Right. So I think one has to take into account the cost that these individuals incur in the healthcare system without a diagnosis. So there's, a lot of people have looked at the cost of the diagnostic odyssey. So these families are coming back every year to see the physician, and they're getting all sorts of imaging and various specialized clinical tests to try to figure out what's going on and what's coming next and all of these things, and because nobody's really sure, and those investigations cost money, and they keep coming back again and again. So when we think about a genome that can very quickly direct you to know what body systems to look at, what tests to run, and what the future holds, that can really scale down the costs for these families, in terms of their overall longitudinal health care. So I think that's a factor that, it's not just a question of, well, what's the cost of a genome test, but it's what's the cost that's going to be incurred trying to find an answer over many years.
Oh, what would you say excites you most about the direction that your field is moving in right now?
I think what excites me most is the massive advances in data sharing that we're really having. Like I spent a lot of my time about 15 years ago convincing labs to submit their data to ClinVar, and we'd worked with the NCBI team to guide the development of ClinVar as a database for sharing classified variants, and came out with guidance that says this is not patient data, it's your interpretation of a variant with the aggregate supporting evidence. So you don't need consent, you can just submit these. And the ClinVar database has been incredibly critical to the diagnosis of patients with rare disease. So that's been an amazing success, but it only goes so far because it just shows you the end result of a lab interpreting a variant. It doesn't actually share the underlying data and evidence. Because if we want to change the classification and build on it, we need to combine the evidence, both the evidence that was there at the time the lab classified it and then new evidence that comes along later. And so that is a more difficult challenge because now you're trying to share direct patient data. And so we've been, developing approaches to do that more efficiently. So a platform we've launched is called the Federated Variant Level Matching Network. The idea here is, if I sequence a patient and find a variant, no one's ever seen it or it's uncertain significance, I could say, well, I just want to query the entire world's genomes, find that variant in a handful of patients and get their phenotype back. And that would help me interpret this, variant in my patient. And so we are actually doing that. We're guiding these labs to develop variant data stores out of their pile of VCFs that they've run on patients or research subjects, and then formatting that data so it can be queried with the Beacon version 2 API that developed in Global Alliance. And so we can query and find that variant in these data stores. And then using the phenopacket standard, another Global Alliance standard, we can return a packet of phenotype information back to the querier and actually share data in ways that can't be supported today because most countries can't send their patient data outside of the country or outside of their institution even. They don't have consent for that, but they can kind of give aggregate information one variant at a time more easily. So we're building that network right now. We have 4 nodes connected and many more coming online. And so I'm very excited about projects like that will actually be able to start to really share broadly, individual level data that's never been shared before.
And then kind of on the other hand of that, what concerns do you have about using AI in genomic medicine?
Well, genomic interpretation is an incredibly labor intensive activity. And so it's really tempting to have it be a place where AI can save us time. And my lab is also, like many others, working on the use of AI to help interpret genomes and variants and genes and all sorts of things. And it's actually incredibly powerful. We can, right today we pay biocurators all this money to painstakingly pull pieces of information out of published papers and manually type them into databases. And now I can just load the paper up to, an AI model, ask it to extract the information for me, and it immediately goes, into a structured form, into a interface. So like, there's definitely major advances that are coming. People can load an entire genome into ChatGPT, ask it to identify the cause of disease, and it does a reasonable job. So there's a lot of excitement for the use of AI in, rare disease and genomics. The problem is that the only way it can make a diagnosis is if the information is out there, accessible to the model. And if that data is never shared and never accessible, it can never learn from it and it can never make a diagnosis. So, that's why, my biggest excitement is kind of unleashing this data and making that data accessible, because that is how that will be necessary for these algorithms to actually diagnose every patient and not just the ones that have the common phenotypes that for which the phenotype is already, in the internet, basically, right?
And do you envision a future where genome sequencing becomes more of a routine part of medical care, or do you think it will still remain uh, a tool for specific situations?
I think it won't be long before every baby, perhaps every fetus, has its genome sequenced. I mean, we're already doing that at some level because NIPT is run on probably 75% of all pregnancies. And so they're getting genomic analysis at in the fetal state in majority of pregnancies. The difference is that that data is not yet being looked at the sequence level. It's being used to detect chromosome abnormalities. It's starting to be used for microduplication syndromes, but it's still a pretty small list of what's being looked at. But in the future, and some colleagues of mine are already launching, a company that's going to be doing fetal sequencing. So I think this is, inevitable, and by doing that at the earliest stage of life, then you have that information that you can use throughout the individual's lifetime. And you will, check it for different things at different stages, or be able to inform the context around a medical presentation that might be related to an underlying, predisposition. So I think we will get there, but the question is how quickly will it become, widely utilized? And there's so many factors of inequity that whether it's insurance coverage or other things that um, lead, new technologies and tests to not be equitably accessed by the population. And so I think we'll have the capability to do this pretty soon, but how quickly it becomes really accessible and used by everyone, still a little bit tricky to predict.
I think on my end, that's all of the questions that I had in mind. So I wanted to thank you again for taking the time to answer some of our questions and provide such detailed and thoughtful responses.
Of course, happy to.
And maybe the last question just to wrap it up on. For students and young researchers interested in genetics, medicine, or just rare disease medicine and research, how would you encourage them to learn or pursue?
Well, one thing I might encourage them to do is to get involved in the Clinical Genome Resource Program. So we enable people to sign up as volunteers, and we train and teach them how to curate, um, and be part of our expert panels where we bring in clinical experts in different disease areas to review the work and the evidence that they've gathered. But the volunteer biocurators are gathering the information and presenting it to the experts. So it's a wonderful way to get involved and have interaction with some of the world's leading experts in different disease areas, but in a way that teaches you about genetics and exposes you to some of the brightest minds. And so I would encourage people, and during their training years in college and beyond, to get involved in the ClinGen program.
Wow, that sounds like a great experience. Yeah, wow. I just want to say thank you so much for all your thoughtful answers. It was